Hello, and welcome to Madison. My name is Joseph Lalli, and I am an MD PhD student in the Donna Whirling Lab at the University of Wisconsin-Madison. Today, I'd like to talk to you about my work investigating sex-specific gene regulation during neurodevelopment, and how a better understanding of this process might help us understand the genetics of neurodevelopmental disorders. So to begin with, there is a well-known link between an individual's sex and one's likelihood to develop neurodevelopmental and psychiatric disorders. Now, while cultural factors undoubtedly play a role, evidence suggests that genetic factors are likely to play a role as well. Now, in a recent study of the UK Biobank, Wigdor et al. Calculated a polygenic risk score to measure individuals' genetic autism risk. And as expected, parents of children with ASD have a higher burden of genetic risk factors for ASD than the general population. So you can see here and here. However, on average, Wigdor et al. Observed that asymptomatic mothers carry more risk for ASD than asymptomatic fathers. This is consistent with the idea of a female protective effect, where females with identical genetic risk burdens are less likely to develop autism than males. It is currently unknown why being female would protect you from developing autism. Now, one way of determining that mechanism would be to learn more about how the same genetic variants impact gene expression differently in different sexes. So to that end, our team is using the BrainVar dataset of 176 diverse human samples. We have performed eQTL studies to examine how individual variants affect nearby gene expression differently in males and females. And this kind of work can then be used to investigate sex-specific regulatory sites that are associated with neuropsychiatric genetic risk. The BrainVar dataset covers the span of neurodevelopment from 8 weeks to 20 years old. However, we are mostly interested in field development, and so our work is focused on these 81 samples. We have paired RNA-seq and whole genome sequencing data from the developing dorsolateral prefrontal cortices of these individuals, who together have 2.3 million variants that meet quality thresholds and 16,000 expressed genes. Now, because there are several different methods of performing quality control on paired RNA, DNA data sets such as these, we have tried several approaches. And primarily, we have focused on performing linear regression of phenotype by genotype in males and females separately, and then comparing the effect size of these genotypes using a z-test. We call this the beta Z method. As an example, here we see neuroplasticity gene DOC3. While the identified mutation R2013-208 does not greatly affect DOC3 across the whole sample, it does have a significantly inverse effect on males and females. This mutation causes an increase in gene expression in males and a decrease in gene expression in females. And we can also perform a linear regression on gene expression by genotype, but this time incorporating sex as an interaction term. This method also works well, and we call this the interaction method. Both the beta Z method and the interaction method have been published in prior work. However, no matter which of these methods you use, it creates 77 million statistical tests. That's a lot, and you're likely to come up with hits at random. So for each gene, we have to correct for multiple comparisons. We first find the most highly associated variant for each gene, and then perform a Bonferroni correction on the p-value of the association. And we do that by multiplying the number of comparisons made for that gene, or we can use the number of effective comparisons as determined by an eigenvector analysis. That takes into account linkage disequilibrium. If you have 500 variants, but 200 of them are always inherited together, that's effectively one comparison. So you can reduce the number of effective comparisons that you're making within that data set. Now, we found these methods yielded a handful of genes that are significantly regulated in a sex-specific manner. Comparing z-scores yielded more hits than looking at an interaction term. And using an eigenvector-informed Bonferroni hit or correction method yielded more hits than using a simple Bonferroni correction. But we also tried a Bayesian approach called SUSY. Now, this work comes out of the Stevens lab in University of Chicago. Now, SUSY, or the sum of single effects, is a method that looks at all variants tested per gene at once. It then goes through the most significant hits first, and iteratively determines if the effect size observed with that variant is best explained as noise, part of a set of variants in linkage disequilibrium that together likely represent a real effect, or as part of a new credible set on its own. One nice aspect of this model-based approach, and I have verified this with the people who wrote the code, is that once the model is fitted, we do not have to perform multiple comparisons correction. The model simply stands on its own. And so that yielded some unexpected results. Specifically, Susie showed us over 100 genes that were differentially regulated in the sexes, only one of which was shared with prior methods. An examination of these sites revealed why. They were due to preponderance of small, credible sets of effector sites. These mutations, all in linkage disequilibrium with each other, clearly together, one of those mutations affects DOC3 in a site-specific manner. But because they are all in perfect linkage disequilibrium with each other, they forced us to use a harsh multiple comparisons correction under the false assumption that all of these variant expression comparisons are statistically independent. And in fact, they are not. So for example, Susie is able to recover this credible set of 4 variants that affect the expression of DOC3. These 4 variants are in a previously identified hotspot of GWAS hits associated with intelligence and income. Finally, gene ontology analysis of these Susie identified E genes shows that the genes that we found to be regulated in a sex-specific manner were previously identified with global developmental delay and motor disorders. In addition, these genes were commonly present near binding motifs for transcription factors associated with cell cycle regulation and neurogenesis. Now, this work is only a start. We're currently working on a next flow pipeline to align our data set to the new T2T reference and to calculate RNA-seq read coverage in an allele-specific manner, allowing us to increase our study's power. We will also be performing splicing QTLs and looking for differential isoform usage. We intend to employ a new tool called MV-SUSY that is able to allow direct comparisons of effect size between 2 groups. Down the road, we'd like to calculate associations between transcription factor binding sites and the location of our credible sets. And we would like to look at using network analysis to find transcription factors that combine to regulate these genes. Now, if you have any questions, you can please email me at lalli@wis.edu. And I'd like to thank you again for your time.